This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 174, recorded on March 9th, 2012. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today from North Central Florida, Rich Condit.
Hello there, Vincent. How are you?
I'm well. How you doing down there?
I'm doing great.
Good.
I'm having a good time.
What's the weather?
My little weather bug is gone from my desktop, but I'm guessing between 75 and 80 degrees. Cloudy. It looks like we're going to have some thunder showers this afternoon, but it's good. It's all good.
It's starting to warm up here.
Good.
You know,
are the crocuses up?
Yeah, they are. They're perfect too, because we've had this weird weather. Last week it was 60 degrees for a day, and the crocuses pop up. Let's see. I'm trying to pick up the temperature. I'm looking at it on my iPad because I'm at home today, and I hate to do this. It's in Fahrenheit. It's 49 degrees Fahrenheit.
Here I got it 78 degrees. That's where I'm at.
They still beat us. You know, whenever I run into people now who listen to TWiV, they always tell me what the weather is.
Sure.
We've started a meme.
It's become an important part of the show, yeah. Absolutely.
Also joining us today from Western Massachusetts is Alan Dove.
Good to be here, and it's 46 Fahrenheit, 8 degrees Celsius.
You ever think of being a weather person when you were young?
No, not really. I knew a guy in high school who was really obsessed with the weather, and everybody figured he was going to go and become a weatherman. I was never that into following it.
He didn't, huh?
I think he may have, actually. I haven't looked him up.
When I was a kid, I grew up in front of our house. We lived across the street from the CBS weatherman.
Oh, cool.
His name was Gordon Barnes.
So did he come out and forecast the weather? When you said hello to him, did he tell you what the weather was going to be?
I never asked him. I felt so weird. He would leave. He'd be gone by 5 a.m. every day. You know, he wasn't around very much. My father used to talk to him, but I didn't have much interest in it. It was pretty cool. But I don't know. Your neighbors, Rich, they live across from the virology guy, you know?
Yeah, that's right. Well, of course, he's an anesthesiologist, so...
Yeah, well, you know, that's the cool thing about living in a university town. And I know listeners were digressing, and you guys want us to cut to the chase, but just...
No, they don't. They don't care. It's all about digression. And if they care, tough.
There's some podcasts I listen to, man, where they start talking about coffee makers because they're drinking coffee. You know what the hot thing in coffee makers are? These little Keurig things where you get the little cartridges with coffee in them.
Oh, yeah, I saw some of those in the store. What am I supposed to do with those? I got to buy a machine, right?
Yeah, you need a machine. And here's the latest thing. I've been listening. They buy this thing that carbonates water for you. So you put a bottle of your own water in and it carbonates it.
Heck,
I have one, and it's awesome.
What's it called?
SodaStream.
That's right. I've heard this. Is it really good?
Yeah. My sister-in-law gave it to us for Christmas, and I thought, oh, okay, fine.
This is absolutely retro. My parents used to have a seltzer bottle, right?
Yeah, right.
It had a CO2 cartridge that you used.
Right, but this one, it takes a CO2 cartridge that's the size of a fire extinguisher.
Oh.
You put this huge, you screw this huge tank onto the back of it, and we use it all the time. We were making our own sodas, and we never really drank much soda before. Now we do, so maybe not a good thing. But
well, you could put flavor in it, but no sugar, I think. Right?
Yeah, mostly we just have like club soda or something where we put water, and it's
interesting. When we lived in Manhattan, our neighbor used to get delivered a rack of seltzer bottles. You know, the old wooden rack with 12 of the old style glass with the metal appliance on top. Remember?
Yeah,
it's incredible.
Okay.
So these podcasts, they're all gaga about Keurig coffee makers and whatever that seltzer thing is.
Yeah. Good thing we never talk about stuff like that.
Yeah, right. We stay on topic.
Right.
Today's topics, by the way, I guess, we said last time we were going to post them, right?
We didn't do that.
I could do it today because nobody's listening right now.
Oh.
But where should I post them? I'm just not sure where everyone's going to catch it,
I wonder if you could, you could put it on the This Week in Virology, Facebook site, but I don't know how accessible that is.
Yeah, I have to be in Facebook.
You could tweet it.
Yeah, the thing is, I don't know if everyone's on Twitter, right? And then I have to do it.
Twitter's publicly searchable. I think anybody who is interested is probably going to be able to find that.
Yeah, maybe I'll do both, and that'll cover it. It doesn't take much time.
Yeah.
All right, somebody asked me what I've been doing. Please, I can't just say.
Yeah, Vincent. You're at home today. Why are you home? What have you been doing?
I just landed an hour ago from Amsterdam. I was there for 24 hours. I left Wednesday night, got there Thursday, and I gave a talk at a small meeting in Utrecht on Thursday. So about a year ago, a TWiV listener, Ewout, E-W-O-U-T, he's an MD who does infectious disease prevention kind of stuff over there. He invited me to give a talk at this little meeting. It's a meeting of clinicians and epidemiology types.
That's cool.
Small meeting, 60 people. He said the Dutch have to hear about this stuff that you do.
Okay.
So
you talked about TWiV?
I talk about TWiV and blogging. I have this talk, and I recorded it, so I'll put it up this week sometime. But
cool.
It's really interesting because people get really excited about this. And the thing they had at this meeting after the talk, people could text in questions, and then the AV guy would put them up on the screen. So if someone's sitting in the audience, they don't want to raise their hand, which sometimes happens, they can just text stuff up, which is cool. So one of the questions they had was, how do you deal with the European listeners who don't speak English? Which is a good question. So you guys have to make your own podcast.
Yeah.
Right.
So I also met another person, Jane, who was Irish, and she works in the Netherlands, and she's a TWiV fan. And actually, I met a couple of TWiV fans.
Cool.
And some of them told me the weather. It's just funny. I think it's really cool. Anyway, thanks, Ewout, for inviting me, and that's where I was. I got back just now and in time for TWiV. That's why I'm home, and if the audio doesn't sound the same, it's because the equipment is totally different. But I was reading the papers on the plane. This is a very interesting exercise. I'm reading them on an iPad. So I have about 6 different applications because I'm trying them all. And I try to highlight. And it's just really variable. Some of them you highlight and you save, and then the next time you open it, it's gone. Others you highlight it, and it highlights a different line. I think it has something to do with the PDF. So
built-in iBooks doesn't, or the free iBooks app doesn't do this?
It might. I didn't try iBooks. Does it? You think it does highlighting? Yeah.
I think so. I've read some PDFs in iBooks, and I'm doing it on a phone, so it's really the problem is the screen size. But I think you can highlight in there.
Do you read your articles on a phone?
No, not because in 2 columns it's impossible. But I've read a couple of things that were PDFs that were single column.
Anyway, so I'm experimenting, and I found a few that I think does a lot of things. As soon as I'm happy with it, I'll tell everybody what it is. But then I upload it I use Dropbox, and I upload the highlighted paper to Dropbox. Now I'm looking at them on my Mac at home, and it's all there.
This used to be so simple. You just Xerox them in the library and then sit in a comfy chair.
I know, but if I'm away, I can't print anything out, right? It means I would have to do it ahead of time, and I really like the ability of doing it on the fly.
No, right. There are advantages. I'm not denying that.
So, and then I said at the meeting, actually, I live tweeted a couple of the talks, so I like being that technology too. So it's all cool.
Absolutely.
All right, today we have 2, I think, really cool papers.
Oh, we're going to talk about papers?
We could just
talk about the weather again.
Yeah, we could probably do that.
Okay.
And our papers are always interesting, but I found these 2 really kind of neat. Let's do the Journal of Clinical Virology paper. It just came out by Suma von Bolsdorf and Maunla. These are individuals from Finland, the Department of Food Hygiene and Environmental Health. The title is Pet Dogs, a Transmission Route for Human Noroviruses? ? And the idea here, everyone knows noroviruses on TWiV because we've had, I think, at least 2 shows on them.
Yeah.
Big causes of human gastroenteritis. RNA viruses, little RNA viruses spread fecal-orally from person to person, contaminated food, water, surfaces, vomiting on airplanes. Did we talk about that on TWiV?
It's the number one foodborne infection, isn't it?
I believe you're correct. Certainly the number one viral foodborne infection.
Yeah, I've seen that factoid stated repeatedly. I have not grounded it all the way back to a reference, but they're certainly very common.
A two-bucket disease.
A two-bucket disease,
oh,
yes.
Oh, we have a good image for this, don't we?
That's right. Alan sent that.
Oh,
the guy, a listener sent it to us.
A listener sent that to me.
Oh, that's right. Okay.
I got it too at the same time.
Yes.
That's a good one for this. Anyway, so these neuros, there are a number of different serogroups of human neuros, but then there are animal neuroviruses, and they're thought to be species-specific in that, animal ones don't typically infect people. But in this paper, they want to know, can human neuros infect pet dogs? And they say there's no evidence of this previously, but there was an outbreak in the UK in a retirement home suggested to be caused by a sick dog. I just have this vision of a dog vomiting all over and these poor people getting sick from it.
Right. So that's pretty anecdotal to go after this.
Yeah.
But
on the other hand, this doesn't seem to have been an incredibly elaborate effort to expend.
No, it's pretty simple.
Right. No, it's pretty straightforward.
Dog poop is extremely easy to find.
Get dog poop.
It's a little too easy to find.
Extract RNA and you do RT-PCR basically.
Right. Because you know what virus you're looking for.
So they used certain criteria. All right. So they got 92 stool samples from 47 breeds of indoor pet dogs. Indoor. It's interesting. Don't dogs go outside? I guess they have to poop, right?
Sure as heck.
Because dogs don't use litter boxes.
Well, and they even say in the paper that they collected all of the samples outdoors.
Yeah, that's right.
All 92 samples collected outdoors, avoiding any other environmental contaminants.
I don't know how you
So they're indoor, I guess, indoor pet dogs as distinguished from
wild dogs. Yeah. So the criteria were that the dog or the people in the household got diarrhea or vomiting. They had a bout of it. So they had one or two where the dog was sick, I think, and then the others, some of the people in the house had gastroenteritis, but they didn't type it as neurogastroenteritis. It was just vomiting or diarrhea.
Now, some of these were controls, right? They had, it looks like they had a bunch of samples from where neither the dog nor the humans were sick.
That's right, asymptomatic, right. I'm glad they didn't have a positive plasmid control.
Right.
Because I was at this talk yesterday, and the guy said, "Don't do positive plasmid controls in the same lab. Do it somewhere else."
Yeah, absolutely.
Really a problem. And so the bottom line is 4 of the 92 samples were positive for a group 2 human neurovirus. All right,
and one of them, they had one sample from a symptomatic individual in the same household as the dog.
Yeah.
And this to me was critical. And that this is the only sample that they actually analyzed from the human. And the virus in the human, as I understand it, was identical to the virus in the dog.
Right.
Right. So the dogs, an Irish setter, a dachshund, and 2 poodles tested positive.
Love it.
And they had another one, a good correlation with kids in the house was good. I'm not finding the data here. All households with neuros and dogs contain small children. They also, all those 4 positives, they had kids in the house as well.
Right.
So of these 4 samples, 2 of them came from situations where there were both sick humans and sick dogs in the same household. 2 of them came from situations where there was an asymptomatic dog and a sick human. But of the 38 samples that were analyzed where the humans were asymptomatic, whether or not the dog was sick, they found no norovirus in the dog. So there's a pretty strong correlation with, if I don't know how strong we're talking pretty small numbers here because we only got some more positives.
Yeah.
But all of those came from dogs that were in association with sick humans.
Right.
And none where the humans were not sick.
They also have a graph of the number of samples versus days after, I guess, disappearance of symptoms. So all the positive ones come pretty early after the symptoms go away.
So it is. They comment on this. It's not persisting in dogs for a long period of time.
Yeah.
Whereas noroviruses can persist in humans for several days or even weeks, I think.
Yeah.
Right.
So,
although, as you said, I think it's important to reiterate, these are very small numbers.
Oh, clearly you have to do more, but it says that the virus can go through the dog's GI tract, right?
Yes.
So they had to probably ingest it to get into its feces.
Right.
So they're thinking, if you have houses with kids, the kids are vomiting all over the place. The dogs are licking the kids or licking surfaces where we know noroviruses can survive for a couple of days. Dogs go outside and they eat all kinds of things, right? They could pick it up outside, I suppose.
And then they lick your face.
Yeah, they point out they can lick your face, or they sleep with 45% of...
he's just being friendly.
45% of household dogs sleep with their owners.
Yep.
No dogs licking my face.
Infection may also occur when owners kiss or are licked by their pets. So, if anything, this is a good reason not to lick or kiss your pet. My son does this, and I said a while ago, there's all kinds of bacteria there, and he said to me, yeah, we have all kinds of bacteria too, but now I can tell him it could be worse.
Yeah.
And so we don't know if it replicates. We don't even know where the dogs in these cases picked up the virus from, although I think there was one case where it was identical. You said, Rich, same
yes,
virus?
Identical to the human. It's the only human sample that they actually analyzed, okay? Because they had a lot of sick humans in this study, but they didn't look at the viruses from the humans except in this one case.
Right.
And in that one case, there was a sick human and a sick dog in the same family, and the virus, they say, was identical.
Yeah. So basically, we don't know if the dog gave it to the human or vice versa.
Right.
It seems likely to me that it was the other way around.
Which way is that?
That it was.
I gave you both ways.
Sorry, yes. One of the above. No, it seems likely to me that the human gave it to the dog.
That's what seems likely to me.
These are human noroviruses.
Right.
And
yeah.
You don't seem to see it, again, it's small numbers, but the ones that you're detecting are very soon after the symptoms in the humans.
And not real high titers, as I understood it.
Yeah, this strikes me as just sort of something the dog swallowed, and maybe it's replicating the intestine, maybe it's not, and then it's coming out.
Mm.
Right.
So the likelihood of the dog picking it up outside is low, right?
Seems to me.
I mean, there's no human fecal material in most developed areas.
This is a developed country we're talking about, yeah. And these are pet indoor dogs.
So the most likely scenario is that the dog transmits it among family members, which would probably happen anyway, right?
You mean family members transmitting it? Yeah. I think the dog's going to be a minor vector relative to the humans.
Yeah.
But I do think someone should do more extensive studies, and as they suggested here, to look for antibodies in the dogs to see if it's replicating.
Mm-hmm.
Yeah.
To see really what the contribution is. So it seems to me that they would be an intermediate host. Maybe if it's replicating in them, right?
Well,
maybe.
It might also be interesting to actually infect dogs and see if they get sick. I mean, we've got sick dogs that you can isolate norovirus from. That doesn't mean that the norovirus is making the dogs sick.
Right.
Mm-hmm.
So,
right. Well, it may even be the other way around.
Yeah.
Wait a minute. You're doing this to me. You mean the dog got norovirus because he was sick?
Actually, yeah. Now that I've joked about that is a possibility. If the dog is sick with something that their immune system is busy with, it might make them more susceptible. But no, I was kidding.
I mean, if the dog got sick from the human noro, that's fine, but that could be. But if the dog has a dog noro, it's not going to give it to the human. We don't typically see that, right?
We know, yeah.
So there are dog noroviruses, I believe. So it's just suggestive. I thought it was interesting that they're there, and people should do more some seroepidemiology, right?
Cool.
Yeah, it's a cool paper. I think they kind of overclaim it a little bit here and there. At the end they say our results demonstrate that pet dogs have a role in the transmission of noroviruses that infect humans.
Yeah, I think that's stretching it. Yeah.
I don't think those results do that. I think they show that dogs are capable of carrying this in their gut. But elsewhere, they point out, we don't know if it's actually replicating.
Right. So the key question is, does it replicate in the dog?
Right.
And if it did, then it's going to shed in quantities that could infect people. I mean, the dog could go next door and play with the kid next door, so
Sure.
It could be spreading it. So I think that's worth finding out.
Yeah, this is the kind of study that, as you said at the beginning, it's sort of a long stretch on the hypothesis. So you do a quick initial study like this, and if you find something interesting, which they did, then the next thing to do is to follow up on that in a bigger way.
Yep. All right. I think that's all we need to say about that.
Yep.
Somebody at this meeting said we talk too much.
What?
They said the essential material is about a third of what we actually spend on any given paper. I'm not worried about that because I think the conversation is part of the whole shtick, don't you?
Yeah.
I mean,
I'm not even thinking about this, right? We're just having a conversation.
Yeah,
your whole model here from the beginning was that we have these conversations in our office all the time, and it's a shame that they're not public because a lot of stuff happens. And it's not just the science that happens, it's the interaction amongst the scientists and everything else. Yeah. So we're just having a conversation.
Yeah, and I think it's true if you want a quick podcast that reviews the latest happenings in virology for virologists so that they can catch up on it. You could probably do that in 10 minutes.
Right.
But
that's not what this is.
That's a totally different podcast.
Yeah, Rich and I are going to do that when we retire.
Right.
We are?
All right, I'll find somebody else. It's okay.
It's okay. I'll do it with you, Vincent.
It's just a 15-minute podcast.
I can do it.
That would actually be a big time. It would probably be a bigger time investment than this one
because you have to be really precise.
Yeah,
you'd have to be.
The talk is harder to write than an hour talk.
And you'd have to be constantly scanning the literature and finding all the interesting stuff and summarizing. Yeah,
I think it would be fun. I'd like to do that.
It could be fun. Yeah,
virus talk,
but that's a different podcast.
Daily virus. All right.
Moving along,
and the.
Right out.
Is very good. Moving along. Could that be an episode title?
Oh, maybe.
Moving along. The next paper is just published in PNAS. Kincaid, Burke, and Sullivan down in Austin, Texas.
Right.
I know this department. Steve Goff was just here. They have a lot of TWiV fans in Austin.
Oh.
So hi, TWiV fans in Austin.
Is this the department that Bob Krug is chairman of?
Yeah, I believe so. Yeah. And he's hired a bunch of virologists. Maybe these are some of them. But Goff came back. He said, you got a lot of fans there. So I said, cool. I like this paper. RNA virus microRNA that mimics a B-cell oncomere. And it's a cool result. And I learned a lot, too, from this, which I didn't know.
This is Kincaid, Burke, and Sullivan.
Yeah. It's PNAS, did I say that? February 2012. Now, everyone knows, I think, that microRNAs are small, meaning 22 nucleotide RNAs that most eukaryotes make, and they're regulatory. I think the human genome has more than 1,000 of them, and they regulate gene activity post-transcriptionally. Either they degrade RNA or they prevent its translation. And a bunch have been found in viruses with DNA genomes. But whether they're an RNA virus genomes is controversial. There are some reports, but if you read the paper, you'll find that not everyone believes the reports, basically.
Right, and one of the problems with that is the processing of mRNAs involves cleaving.
Mm-hmm.
And so if you had an RNA virus that encodes mRNAs, these microRNAs, and they're going to get cleaved, then that raises an immediate problem. How do you keep your genome from getting chopped up?
Yeah, you don't want to cut up your genome. And so the ones in DNA that are DNA-based are made as Pol2 transcripts, and then they're processed by a ribonucleoprotein complex processing complex down to the 22 nucleotides.
RNA polymerase 2 transcribes these, and then they're processed down.
And we had a transcription TWiV, right?
That's right.
That's right. We did.
We referenced that.
So there's 3 different RNA polymerases. RNA polymerase 1 makes ribosomal RNA. RNA polymerase 2 makes messenger RNA. RNA polymerase 3 makes a whole bunch of other small RNAs, including tRNA and 5S RNA.
Mm-hmm.
A bunch of other guys.
Right.
But Pol2 usually makes microRNAs, right?
Right.
Or makes the precursors.
Makes the precursors, then they're processed by Drosha in a complex to chop them down.
And then further processed by Dicer.
Dicer too, right. Now, there is one, the reason they did this paper, this study, is there's one DNA virus, a herpes virus, murine herpes virus 68, which has microRNAs which are made by Pol3. It's a different polymerase, and they call them tRNA-like precursors. And so they thought, well, Pol3 is made from a DNA genome, and they said, well, let's look and see if there are any Pol3 made microRNAs. So they made an algorithm to look for these. So basically, they looked for certain kinds of small sequences that would be made by a Pol3 promoter. So the algorithm is based on the promoter. They scanned all the viral sequences, and they picked up a couple of them, but the ones that they focused on in this paper are from bovine leukemia virus.
Right. So this is a retrovirus that, so you got an RNA genome that makes a DNA copy that integrates into cells and then expresses genes and ultimately makes an RNA genome copy that's packaged and transmitted. And this is a virus that induces B-cell lymphomas in cattle.
Yes.
And people had suggested that if you were going to find mRNAs in RNA viruses, retroviruses would be a good candidate for that because they have this DNA intermediate and could therefore potentially survive or otherwise get around this cleaving of the genome problem.
Right. Actually, I don't. In some ways, it's not fair to call retroviruses RNA
viruses. Right.
They have RNA genomes.
That's what they package, right?
Yeah, they package RNA, but heck, same life cycle, you can package DNA and you're a hepadnavirus.
Hey, this brings up a good digression. So when I was teaching the reverse transcription lecture, I was talking about, so there are a number of viruses that have reverse transcriptase in their life cycle. There's retroviruses, hepadnaviruses, and then these foamy viruses. So the retroviruses package RNA, and then when they infect the next cell, they reverse transcribe it to DNA. The foamies and the hep Bs package DNA, and Steve Goff said they package DNA because first they package RNA, and they can't wait to reverse transcribe it, and they can't control themselves, and they just reverse transcribe it before the particles even leave the cell, and that's why they have DNA genomes. Yeah. So it's basically the same strategy. It's just where you do the reverse transcription.
Right.
So calling the hepadnas DNA viruses, they could be RNA. But the spumas, the foamy viruses, actually package DNA, but they're called retroviruses, right? So these guys found these sequences, then they synthesized them. They put the candidate microRNAs in the plasmid, and they found they made microRNAs in cells. They found they sequenced them and showed that they were the right predicted microRNA. And then they looked in virus-infected cell. They have a cell line that's, I guess it's the genome of the retrovirus is integrated. They produce particles, and these microRNAs were there as well in these cells. So they're bona fide Pol3 microRNAs. One thing that I really was amazed by is all these northern blots where they're looking at 10 to 70 nucleotide RNAs, and they're probing them with oligonucleotides.
Mm-hmm.
Because when I used to do northerns, it was always big stuff, and stuff this size would be at the bottom, and we'd ignore it.
Mm-hmm.
It's amazing. And they can tell what's made and what's not. Now, the cool thing that comes up here, all right, before we go on, it was one cool experiment that I liked a lot, and they wanted to prove that in the infected cell, these were Pol3 transcripts. So they add alpha-amanitin, which is a drug from a fungus. It's the one that kills you, that fungus, right? Amanites phalloides, a mushroom. And it does not inhibit these microRNAs, which shows that they're not Pol2 transcripts. I thought that was pretty cool. Right. Now, it turns out that these, when you get a microRNA, you want to know what its target is. Right, and it turned out that the seed sequence of this microRNA, which is really important for hybridizing to the target and causing degradation, is very similar to a couple of cellular miRs, miRNAs, that have been implicated in oncogenesis of B cells, so the production of B cell tumors. And what their targets are is how that works in oncogenesis isn't clear. So in other words, what are the two? miR29 is one of these host miRs that hits.
That's the major one.
And overexpression of that transforms cells. It does something to cells to make them transformed along the way to a B cell tumor. And so these viral ones are mimicking that. So this could be the mechanism of oncogenesis by BLV.
Right.
And they show experimentally that if they that the miR29 targets and the targets of this thing are basically the same, or they can be the same. So if they stick a miR29 target on an RNA, that can be downregulated by both miR29 and this bovine leukemia virus microRNA.
Yeah, I'm looking for these. So there were two targets here that they looked at.
Yeah, it's HBP1 and PXDN.
Right.
And I forget what they do.
So these are messages. These are genes, right?
Those are genes that are targets of the miR29, the cellular microRNA. And they can do this thing where you put the target sequence on a luciferase reporter, and if the microRNA targets, and then do a transfection, look for luciferase expression. You transfect in both the microRNA and the luciferase reporter with the target sequence on it, and if the microRNA targets the luciferase transcript, you get less luciferase activity out the other end. And they show that miR29, the cellular guy and the leukemia virus guy, both get these, target them.
Yeah.
Now, what I forget is exactly what those genes do, if they even know.
I don't think they know how they participate in transformation.
Right. I think that's still a mystery.
Yeah. But if you overexpress miR29, it drives them to hyperproliferative disorders. In other words, they keep dividing, and that's the precursor to oncogenesis. And they also find that miR29 is overexpressed in a subset of mild human chronic lymphocytic leukemia. So this is clearly involved in some forms of transformation, and how it is, it's not known. But the fact that the bovine one does it makes sense because, again, the tumors apparently all have integrated genomes, bovine leukemia virus genomes, and so making this paltry transcript would fit.
Right.
Perfect sense. So I
guess in this bovine leukemia, it's not so much that the B cells are multiplying out of control, proliferating out of control. It's that they don't turn over as fast, so they live longer.
Right. That's right.
So they accumulate.
Yeah, start to accumulate mutations, right?
Which is it's similar to one of the forms of chronic lymphocytic leukemia in humans.
Yeah.
And so basically making your host proliferate is an advantage for the virus.
Right, or preventing it from dying.
Right.
Yeah, that's right.
So the virus, as often happens with oncogenesis in viruses, it's just incidental to the virus trying to preserve itself.
Yeah. So the oncogenesis happens because the cell accumulates mutations as it grows uncontrollably. The virus doesn't care about oncogenesis. It just wants the cells to divide.
Right.
Now, here's a cool question. So so the paltry transcript is produced from the proviral DNA, right, in the cell. It's infected with the virus. It is processed. It is not actually. They show it's not processed by Drosha.
Right, and that's an important thing because remember this thing we talked about before is that theoretically it's not a good idea to have an RNA virus have a microRNA in it because it basically is putting a processing site in the RNA genome.
Right.
But these are configured in a fashion so that they are not processed by the normal pathway, which would be Drosha.
Right, because if this were in the DNA, it would also be in the RNA, and then you would say, why isn't the RNA cleaved? And because the RNA is not cleaved by Drosha. But the actual transcript, the paltry transcript, folds in a way that I guess is not possible when it's part of the full-length RNA genome. Because remember, it's a tRNA-like structure, so I have a feeling that it has to be pretty short to fold that way, and so that just doesn't happen in the RNA, the mRNA of the virus. Does that make sense?
Yep.
So I think that's pretty cool. It's a nice evolutionary twist, to avoid chopping up your genome.
Yeah, and the another thing I noticed is that the miRs that they find here seem to cluster. Either they were all put in that part of the genome for some reason, or they just were all inherited at the same time.
Yeah, and in fact, they cluster in a part of the genome that doesn't code for proteins.
That's right.
Which is also useful.
And I was a little confused about how that, because there's got to be some processing. But I guess they all have Pol 3 promoters and terminators. So they must just go straight to dicer and get processed. And just skip the whole drosha
They
thing.
skip the drosha, right.
Right.
They are processed by dicer, right.
Okay.
So I think this is probably opening up a whole new area because now people can look for these Pol 3 transcripts, and they say here it opens up a role for non-coding RNAs in tumorigenesis and suggests that other retroviruses exist that use non-coding RNAs in their infectious cycle. So it could be really interesting.
Right. And right at the beginning, they allude to what seems like a big controversy about microRNAs in HIV.
Yes. And Steve Goff has told me the same thing because I asked him once, are there microRNAs in retrovirus? He said, everybody's arguing about it.
Yeah, so this suggests a way that you might go and look for those now that might help settle that.
I wonder if there are such microRNAs in eukaryotic genomes. Why not, right?
Pol 3 transcripts.
Sure.
Could be.
Yeah.
Why not?
I don't see why not. I mean, it wouldn't be necessary. I mean, it could be part of a coding region where you don't want to clip up the mRNA, so you would put a Pol 3 promoter in there. And
sure.
Oh.
Actually, I think this is one of the coolest things about this paper is it all starts with a bioinformatic analysis. I was pretty impressed with the fact that they could come up with an algorithm that could fetch out Pol 3 promoters and then use that as a screen to look for these things. As well done.
Right, and then bootstrap it into finding more of them.
Yeah, it's very good. I also like the idea that they took the lead from this herpes virus 68, which made a Pol 3. They said, maybe there's some others. Let's look. That's just really nice.
Yeah.
Now, that virus, of course, is also biologically relevant here.
Which?
MHV 68.
Right.
Because this also transforms something that's...
It causes B-cell lymphomas.
B-cell lymphomas. And that has a microRNA that may be implicated in that process.
I believe so, yes.
That's the idea, yeah. So it's pretty interesting.
Yeah.
And Brian Cullen has a nice commentary, a one page. I don't know if it's open or not, but we'll put the link up in case it is. But it's really nice summary.
Yeah, I think these are both behind the paywall.
Too bad.
Unfortunately.
Yeah, okay. Cool paper. All right. Anything else on that before we move on?
No, that's good.
Good. All right, let's do some email. I think Alan should read the first one.
Sure. Yes, it's to me. So Ian writes regarding Alan's query of rectal swabbing in distinct lineage of influenza A virus from bats paper. Wouldn't the rectal swabs have had more to do with looking for the possibility of transmission of a virus via the guano, with human exposure to the guano being collected for use as a crop fertilizer being the theoretical infection route? That's a good point. That might have been the reason for swabbing that way. It just as I read it, I thought, wow, it's a mammal. I would have gone for the respiratory route. But yeah, if you're approaching this from a public health standpoint, that does make sense.
Mm-hmm.
People are exposed to more bat poop than they are to bat sneezes. I would hope.
I guess if you go in caves, you would be, right?
Yeah.
But just in everyday life, I mean, it depends where you are, I suppose, right?
Yeah, and bat guano is used as fertilizer.
That's it, yes, of course. But I don't know if the process would inactivate the virus or not. I just don't know. The next one is from Jason, who writes, "Hey, TWiV crew, I took the opportunity to donate blood today and noticed that in addition to the normal information about HIV, hepatitis, NAT, etc., testing, there was a page and a half of information about XMRV and CFS. I pointed out that the information they provide has effectively been debunked to the first screener who effectively dismissed my comments. I think they were volunteers and not really medical professionals, so that response seems normal. When I was then screened by an RN, I mentioned the problem again. She seemed taken aback that I would claim their pamphlet was incorrect and then effectively dismissed my claim. In looking over the pamphlet now, I noticed that it's the 2008 revision, so I suppose the data on there is accurate for about that time frame, but I find it somewhat irresponsible to continue spreading incorrect information so many years later. I do find one of these questions somewhat amusing. They ask whether a person diagnosed with CFS should be donating blood. The answer they provide is that while the person donating should be in good health, it's up to the medical directors at the blood collection centers to decide whether or not people diagnosed with a history of CFS should donate or not. With all of the precautions prohibiting donations from people who have been in contact with others who are diagnosed with illnesses like hepatitis or HIV, I find it hard to believe that they leave it up to the medical director to make the call on CFS. At any rate, keep up the interesting podcast. Despite not being in the field, I find it stimulating and I'm learning a lot, or at least learning enough to cause trouble. Yeah, right.
Yes.
Keep it up. So through my work with CFID's organization, I happen to have met Roger Dodd, who is Vice President for Research and Development at the American Red Cross. So I sent him Jason's email, and he said, "Thanks for asking. This is an interesting commentary. My first comment would be that while most aspects of blood collection are highly standardized, different organizations may have different approaches to certain issues that are not defined by regulation or voluntary standards. Certainly management of CFS and XMRV would fall into this particular category. AABB, the Professional Organization for Transfusion Medicine, did issue some guidance to its members in 2010, recommending that blood collectors educate donors and ask them to refrain from donation if they had a medical diagnosis of CFS and providing website information for CFIDs. Interestingly, and to my mind appropriately, XMRV was not explicitly mentioned. For some blood collectors, the Red Cross is one. This procedure is still in place. Red Cross materials, however, do not mention XMRV. However, the task force that originally made the recommendation has advised AABB that its members should revert to whatever practice was in place prior to the recommendation. It is possible that the educational document was handed out along with other materials dated 28. I doubt that any blood organization in the U.S. had any explicit materials about CFS in 28, and they would definitely not have cited XMRV then. Blood collection staff are supposed to be knowledgeable about the materials that they distribute, but they are ingrained in a highly disciplined and strongly regulated environment. If they have not been retrained, it is quite possible that they would adhere to prior requirements. Unfortunately, it is also possible that they would not be particularly knowledgeable about the medical and scientific issues at hand. Finally, in areas of medical uncertainty, it does fall to the medical director to make final decisions about donor eligibility. The overriding criterion is that the donor should be healthy and feeling well at the time of donation. Your correspondent has asked astute questions. I hope you can make sense of my responses. Please do not hesitate to get back to me if more is needed. If you would prefer the one-word answer, it is inertia. Well, thank you,
That's
Roger.
really great. That last bit. Yes, inertia.
I could fully understand that people who are screening, they wouldn't know anything about XMRV.
Well, and also, cut them some slack. These are people who are dealing with the general public, many of whom have some very, very strange beliefs. And so one more person coming to them and saying, hey, this has been debunked. Well, maybe not, but that's what the that's what the pamphlet says, so they're going to stick with it.
Yeah, this is the procedure. So
yeah.
Whether or not, if you have CFS, you should give blood or not is an interesting question because it may be an infectious disease, but who knows if it's a trigger and then the infectious agent goes away, and you feel badly, but you could still give blood. So I don't know what the answer is there. That's a whole different issue.
Yeah, and my understanding of the initial call on that when the XMRV thing was still in full flush was, hey, we don't know how this story is going to end, but if this does turn out to be a new human retrovirus, whether it's causing anything, wouldn't you hate to be the administrator who said, no, it's fine for these people to still donate blood?
Yeah.
And so they didn't want to get caught in the same situation that could have happened with HIV, say.
All right. Rich.
Keith writes, Dear TWiVidae, is that how I should pronounce that?
Yeah.
Or TWiVidae. I love that, by the way. We may not be geniuses, but at least we're a genus now.
Yeah.
TWiV is a family, though.
Oh, that's a family. You're right.
But we are a family.
We are a family.
Oh.
My name is Keith, and I am with the HIV Reference Laboratory here in the Bahamas, and would like to know if you have heard are any online PhD programs where the thesis can be completed at one's own lab. If any of you are ever in the Bahamas, send me an email and I could show you around. Okay, so I guess he's asking, so he's in a laboratory and he's asking basically if he can have a remote mentor and in an online PhD program.
Yeah.
I guess that's the idea. I know of nothing like that.
No, I don't either. I mean, you can do this via collaborations, right?
Right.
But I don't know of any online situation that organizes it in any special way.
Yeah,
not any legitimate ones.
Yeah, right. To me,
I'm sure there's some operation out there that offers an online PhD, but please don't do that.
Yeah,
right.
No, if you can find a collaborator that your lab can propose some kind of interaction. The thing is, if you're going to get a PhD from an academic institution, you usually have to fulfill the requirements of that institution, which includes exams and courseworks and so forth. So it's not straightforward.
Yeah, you'd probably have to go to the institution that's granting the degree, take the classes that they require for their PhD candidates, take a qualifying exam, and then maybe go back to your lab and do the other part of the collaboration there.
Yeah, and even then, you're really, for a credible PhD program, both in the laboratory and the classroom, it really requires a lot of face time with the faculty running a program. It would not be a good idea to do it in isolation, I don't think.
No.
However, we could go to the Bahamas.
Yes, although the second part of your letter, I think, we'll all be in favor.
Yeah.
All right, Alan, I believe you're next.
Sure. So Benjamin writes, "Howdy, hosts." That's a good way to put it. I'm one of those 3 high school geeks who listens to TWiV, TWIP, and TWIM. I'm 16, and while some of the subjects, like the molecular biology of zinc finger, are above me, your science is down to earth and understandable. I've been latently infected since late 2010, since TWIP and TWIM had fewer episodes to get a catch-up hold on. A couple of weeks ago, however, it became a full-blown clinical infection and got the world's biggest TWiV fix. What a powerhouse, you guys. Now, thanks to TWiV, I've fallen behind in my other podcasts. I'm glad you didn't say your studies, because then we'd feel really bad. I've had an unofficial game going with Alan since TWiV 20 to see if I can come up with any bad virus puns that he missed. So far, I've only gotten one. In TWiV 58, Nipah virus in ferrets, the scientists doing the experiments were ferreting out the mode of infection. I think I hit that one in a subsequent episode, though.
That's credible.
Yeah. It's no secret that I have a droll sense of humor, so maybe other listeners can go through the TWiV backlog to find more. I would like to offer my take on the aliveness of viruses. Every time you argue the point of viruses, you bring up the prions and transposons. Transposons are not alive since they're genes already in the genome, rearranging themselves spontaneously and stochastically, as far as I can tell. Thus, they are mutations, not organisms. Prions are misfolded proteins. CJD, Creutzfeldt-Jakob disease, and Kuru are basically the protein affecting the tissue and misfolding more proteins. The damage could probably be replicated by injecting pepsin, trypsin, demyelination factors, which are also proteins, and thus prions are also not alive. Then we come to viruses. Viruses are beautifully designed to do what they do best. The characteristics of life list, Bio 221, Earl Bayer on iTunes University, Talaro's 7th edition of Foundations of Microbiology, are many, but several include reproduction, check, a genome, check, and a cell barrier, check. By this reasoning, viruses are alive despite a marked lack of things like irritability. That means response to the environment, metabolism, cellular design et al. Okay. Yeah, I like that.
Sure.
Viruses do meet a lot of the criteria, and I think, I think we've indicated that this is a sort of a permeable barrier between living and non-living. That there are some things that are probably on the cusp there.
Right, and I've always I've never really thought of prions as alive. I guess I'm thinking that it's got to be information transfer in the form of a nucleic acid. I don't know if I can really justify that, but where I get hung up based on my definition of life, which is mostly, as I said, persistence in nucleic acid sequences through time, is that transposons wind up being alive. But he makes a nice distinction here between transposons and viruses that's worth thinking about.
Yeah.
Viruses are not alive. The virus-infected cell is alive. That's mine.
Okay.
Right.
Okay.
By the way, people are still taking that survey. I looked at it.
It's over 3,000 something.
Yeah.
And people still leave comments. Yeah.
Yeah.
And some people get into big arguments and call each other names there.
It's the internet.
Yeah.
All right. Continuing with the letter. I think I tipped my politico-religious hand in the previous paragraph, but that needs tending to as well. I can't imagine that me and one of my friends that I hooked on TWiV are the only Christians, shall I say non-evolutionists, getting our TWiV fix every Monday morning. So in the future, could you please think twice before shamelessly trashing religion?
Did we do that?
We probably made a few comments here.
Probably did here and there, and if so, I apologize.
Yeah, it only reflects our... It doesn't mean that you can't do it, right? So, but we will try not to trash things.
We'll be nice.
Although I will point out that Christian does not necessarily correlate with non-evolutionist.
Right.
In fact, that's a minority of Christians worldwide, particularly if you count the Catholics.
Yeah, I could go on about this for several hours.
Yes, we could.
That's a different podcast.
Yes.
I read this letter yesterday sometime and have been processing it ever since.
Yeah. But you have a number of years, Benjamin, to figure out how you stand on evolution and religion and all that. So
yeah. Well, when I was 15 or 16, I think I was still thinking about going into the ministry.
Okay.
There you go.
Which one?
Different podcast. I probably would have been Protestant.
Oh, okay.
I was raised Lutheran.
Yeah, we'll do a podcast on it one day.
Sure.
Get what's his name, the head of the NIH.
Oh, right.
Francis Collins.
Yeah.
Okay.
In TWiV 150, Rich talked about Buda, Texas. I happen to have lived close to there, by Texas standards. It's about 100 miles, for all my 16 years, and I have never called it anything but Buda.
Yes, I pronounced it Buda, which is what I was told by my daughter it was pronounced. But I'm going to, my daughter is not a native Texan, and I would be overjoyed if it was actually Buda, Texas. So I'm going with that, and I'm going to have this conversation with my daughter again.
And I like that he's an outspoken Christian right next to Buddha.
Yeah.
Anyway, continuing, I've written you on TWiP before about a syndrome that I call stumbling poultry disease. Maybe with all the resources of TWiV, we can get an answer. Description: it only happens in the summer when the local temperatures soar as high as 115 Fahrenheit in the shade. I'll take up Rich on the challenge that no one but a Floridian could survive the Florida climate. Superheating the waterers to well above the required 90 degrees Fahrenheit for E. polyphaga, despite our best efforts. Add that to the fact that chickens and turkeys are sloppy drinkers, meaning that water runs into their noses, and voila, we have poultry displaying weird behavior followed by disorientation, stumbling, vertigo-like symptoms, and eventually death. Perhaps Alan with his Insta-Google or Rich with first-hand experience with the southern climate can add some ideas to the pool.
So we read something about this on TWiP, and Dixon had no idea what it was about.
What I found, I didn't find anything infectious, but I did find some news articles about temperature problems in the poultry industry. And 115 Fahrenheit, which is... what is that in Rankine, Rich? 500 something.
I don't know. I can relate to that in Fahrenheit.
Yeah, so
stinking hot.
That's really, really hot. And Texas hot, that's going to be hot and dry. So there's going to be a dehydration problem. I did find a news story from just last summer when they had those horrible heat waves throughout the Midwest and the South. And the news story talks about temperatures around this range, 110 to 118. and how all the turkeys and chickens were keeling over. So that is, I think at that temperature, probably what you're looking at is just hyperthermia. That's my guess.
Okay. All right. Well, the audience of TWiV, if you have any ideas, weigh in.
Yeah.
I'll tell you, I'm stumbling around when it's 115 degrees.
And the other thing is, at that temperature, you should check to see if those little thermometers have popped out.
The ones on the back of the chicken, right?
The ones on the chicken. Okay, wrapping up the letter. That's about it for now. Love the podcast. Hoping for TWiB and TWiF. Live long and podcast. Greetings from South Central Texas, the summer residence of the golden-cheeked warbler. Cue impromptu ecology lecture by Dick.
Right.
Yeah.
Wow, that's what a wonderful letter, and
Excellent.
I love getting letters from people who are like 16 years old, high school geeks. It's great. So drag all your friends into this and spread the good word.
Yes.
This is who we want to be talking to.
Yeah. High school geeks.
Cool.
That's who I was. So.
Yeah.
The next one is from John, who writes, Dear Vincent, Dick, Rich, Allen, and the TWiV gang, I found the recent experimental use of viruses which can only reproduce in cancer cells, reported in TWiV 156 and 131, as anti-cancer agents to be exciting news. I wonder if it's possible to use natural selection to produce viruses, or for that matter, immune system evading parasites with improved cancer-killing properties, alleviating the need for rational drug design. All right, so my response is that because the cancer cells have defective innate responses, they are efficiently killed by certain viruses, and I don't know if you could get better than that, right? Because they kill them rapidly. So by passing the viruses in cells to select something that kills them faster, I don't think you could do it. What do you guys think?
I mean, with apologies to Benjamin, anytime you're trying to compete with what evolution has already done, it's an uphill battle.
But that would be the experiment. Passage the viruses on cancer cells in culture or in tumors in animals or something like that. And I don't know what you would get, but as Vincent says, if you're already starting with something that kills pretty effectively, I don't know. Maybe you could improve on it. We'll see.
Well, and you can in the lab evolve things that do a particular thing more effectively. You usually pay some cost in selective terms for doing that, and it's not as good as something else. But in this case, the exploitation of the innate immune response is something that viruses have been working on for nigh on 4 billion years. Mm-hmm. I mean, this is a fundamental thing for viruses. It's something they run up against all the time. So it seems like an area where you're probably not going to be able to do a whole lot better just by evolving the virus. But might be worth a try.
So what he's referring to by rational drug design is putting things in the viruses that get delivered to the tumor cells.
Right.
So we talked about that in 156. These armed and targeted pox viruses, put in.
Yeah.
But that seems to be working well. I don't know. I just think you can't kill any faster than a good rapid acute infection. So the advantage of that would be limited.
And I think the engineering approach is probably the way to go, because that's something that viruses would not have been able to do.
All right, the last one, Rich.
Vincent, you're fading in and out there.
Yeah.
Are you okay?
Yeah, I'm fine.
Okay.
You hear that? Is that good?
Yeah, you're okay now. You got a little fuzzy there for a minute.
Okay.
Hi, Vincent, Rich, Allen, and Dixon. I am a software engineer at Google with a bit of education in genetics and computational biology. I've been listening to TWiV and really enjoying it for a couple of years now, but it makes me really curious to better understand the patterns of infection in my own home. It drives me nuts that we can understand so much about viruses in general, but when I get a cold, I don't know what virus's strain it is or where I likely contracted it from. I can imagine a future where it's routine and virtually free to sequence someone's virome whenever they get sick. Imagine what we could do with all that data. So, I want to try and learn more about the patterns of infection in my home. For example, what viruses do we get? Do my kids tend to get viruses from me or vice versa, etc.? I've been reading a number of papers and searching for services/tools I could use, but it's tricky to get a good picture about what techniques would be practical and safe to do myself at home. Perhaps the ideal approach would be to find a lab that would do multiplexed RT-PCR on nasopharyngeal samples when I send them, giving me the viral load for the most common URT viruses. Maybe I could do a little home DNA purification and send it out for sequencing when I wanted to know more about the precise strain. The simplest approach looks like it would be in order to order some ELISA kits. But it would be nice to have the sensitivity and quantified result of PCR so I can track viral load over the course of an infection. I'm willing to invest a bit in lab equipment and services, but would ultimately like to find something scalable and cost-effective. Is there anything you can suggest? Let me finish the letter and we can come back to it. Along these lines, here's a potential pick of the week for you. And it's a link to Biopunk, Do-It-Yourself Scientists Hack the Software of Life. I like how this book relates the current state of biology to the early years of computing and suggests that what it might be possible if biology gets the equivalent of an open source movement and personal computers. Thanks. Keep all the great work on TWiV. TWiStar. That is all the TWiZ. Okay. So, a lot of these analytical techniques that he's talking about are going to be hard to handle in the home. And I don't know of any, it's probably going to be expensive to send it out. I don't know that you could find a lab that would do that routinely. The only thing that I was able to come up with, and I don't know if this is really accessible, is that at least for some of these things, there are rapid diagnostic tools that physicians use. Like I think that you can, there's a sort of a dipstick for flu. I think you can spit on a dipstick and find out if you've got influenza A or B. I don't know about other things, probably not common cold viruses. I don't think there's many of these, but I couldn't come up with much else.
Yeah, there's actually, in the pipeline, there's a whole generation of these things. They are initially being targeted to doctors, and they'd be in-office tests, rather like the rapid flu swab. There's already a rapid strep test, a bunch of others, where it's a quick either immunological or in some cases now DNA hybridization-based test. There's a lot being done now with some of these microarrays that we've talked about on various episodes, which gives you a very, very sensitive, fast test and fast readout. All that is still at the development stages, though. Or a lot of it is still at the development stages. And in terms of deploying it routinely at home, it's going to be, right now, it's going to be a little tough to do that. The RT-PCR approaches are going to be extremely susceptible to contamination. Mm-hmm. We've talked about this on TWiV. It's a problem in the lab. It's going to be an even bigger problem in a lab that you build in your garage, because you're going to have to do your positive controls in the same room. And then you also have the metagenomic type studies where they just look at all possible viruses in a sample, and we've talked about some of that stuff, but I don't think you want to go down that road because the noise is just too enormous. So you would want tests for specific things. Those are in the pipeline, but I don't think there's a straightforward way that this could really be done on a routine basis at home yet. Give it 5 years.
I wonder whether it would be a good thing or not to have over-the-counter good tests for bugs available to anybody who wants it. On the one hand, you might eliminate a lot of doctor's visits. On the other hand, you might generate a lot of doctor's visits. I don't know how that would work.
Well, if you do it at home, you get a result. You go to your physician. He or she will repeat the test with whatever they have.
True enough.
And then decide what to do. But if you don't have an antiviral, it's not going to be worthwhile, right? So that's the limitation to getting antivirals against many acute viral infections. You don't have a rapid diagnostic. So, I mean, once those come together, then you'll have more antivirals. But now,
and over the counter diagnostics are always controversial because you have this whole problem of what are people going to actually do with that information, and are you really just catering to hypochondriacs?
We're going to have a tricorder one day. You're going to pass it over yourself. It's going to tell you everything about you, including what's infecting you.
Terrific.
It's just not here yet. This home PCR thing, there was an article in Wired a couple of weeks ago. You guys may have heard of it.
Yeah.
Where there's a company developing PCR and other approaches for the home use. And you can actually buy these already. And Laurie Garrett referred to them in the New York Academy Symposium about H5N1 a couple of weeks ago. And she and Mike Osterholm are afraid of this whole movement of do-it-yourself biology. I find, having known how hard it is to do experiments in a really well-equipped lab, I'm not too concerned. Although Arturo Casadeval said if someone really wants to do something, they will be really inventive. So I don't know. From my point of view, I think it's really hard to do any of this stuff. You, Rick, might want to get one of these PCR machines, but as Alan said, if you do a positive control, you're going to get contamination because in the lab you're supposed to have different clean rooms in which to do this. You're going to have a whole set of pipettes and PCR machines for your controls versus your samples.
Which has that's become more of a problem in recent years than it was when PCR was first available because the analyses have become so much more sensitive.
Yeah.
I mean, we used to do PCR positive controls in the same tube, or not the same tube, but the same multi-well plate.
Yeah.
And that was just commonly the approach because you were looking at something that was 9 orders of magnitude over background, and so you didn't really worry about the contamination that was inevitably going to be there. But now that we're up to the point of real-time RT-PCR, where every cycle you're getting a readout on exactly what's going on and you're counting individual molecules, now this contamination becomes a huge problem. And I think, absolutely, go ahead and set up a home lab and play around with this stuff. I really encourage it. But as far as the fear that's being drummed up about what people are going to do with this, I think that's based on a false analogy. I mean, the idea is, you have the personal computers, and now hackers can get a hold of a personal computer and get onto the internet, cause all sorts of problems by designing malware. And that's all true, but that's in a system that's entirely man-made. And entirely comprehensible because it was made to be used by people. People built it. When you're dealing with biology, it's an entirely different game. The idea that somebody's going to be able to set up a lab in a garage and synthesize some deadly pathogen that'll wipe out humanity is just not credible. Because that's already, nature has been doing that for as long as we've been a species, and we're still around.
Not only that, but the first thing you learn when you start working in a laboratory is that this stuff's really hard.
Yes.
It doesn't work most of the time under the best of circumstances.
Yeah. I tell you, it took me years to get a polio genome cloned.
Jeez.
And I thought I was pretty good. And I had good labs. So Carl Zimmer wrote an article in the Times recently about this whole issue. Amateurs are new fear in creating mutant virus, which raises a lot of these issues. And they quote Mike Osterholm and Arturo Casadeval, who are worried about this. I'm not particularly worried, and Alan and Rich don't seem to be either. And they quote Ellen Jorgensen, a molecular biologist who is president of GenSpace, a community biotech lab in Brooklyn. She says, I am really sick and tired of folks waving this particular red flag. So what is this GenSpace? I've never heard of that, a community biotech lab.
It's it's based on the concept of hacker spaces, which are rented space where electronics hobbyists can get together and play around with electronics and computers.
I see.
And so people have now started firing these up. There's one in Brooklyn. I think there's one out on the West Coast as well.
Yeah, I think we even had something like this as a pick a while ago.
Maybe.
Yeah, so the idea is it's a basic biology lab, a basic molecular biology lab that people can join. I think they charge a membership fee, and you join, and you can learn how to use the equipment, and you can go ahead and use it. And you can go on eBay and buy a used PCR machine pretty cheaply.
So here's their website: New York City's Community Biolab. Yeah. GenSpace is a nonprofit organization dedicated to promoting citizen science and access to biotechnology. Since 2009, we've served the greater New York area by providing educational outreach, cultural events, and a platform for science innovation at the grassroots level. Looks cool.
Well, maybe, Rick, you can find a similar thing out wherever you are.
Good idea.
And you can bring your samples and do it in a reasonable lab environment. Yeah.
Outstanding.
And in fact, if you can't find it, start it.
Yes.
Find like-minded individuals, and you're at Google. I'm sure you have no problem finding things. So track down people in your area who are interested in the same thing, and get together and set one of these up.
Yeah, do it in a good lab. That's a good idea, not at home.
I wonder where at Google he is, because Google is everywhere.
And do Kickstarter to raise money for it.
Yeah.
Rick, if you're in Seattle, look up my son. He's got the same last name.
As you, right?
Yeah. As me, right.
I will, that's a great idea. I was going to say, I'm going to put a link to the Wired article, which has an article about this company that's making home kits, but you should just do it
and let's put a link to the GenSpace as well, just to keep that idea hot. Right. That's good.
Yeah, I think this is a great this is a great movement, this biology hobbyists.
Yeah.
It's like in Victorian times.
And it makes no sense to let fear squelch that enthusiasm.
Exactly.
By the way, speaking of fear, I was in the Netherlands, which is where Ron Fouchier works.
Ah, yes.
And you're still alive.
Yes. I asked everyone, what do you think of this? And they say, it's not a big deal. It's been in the newspaper a few times, but we don't worry too much about it. He knows what he's doing. He has a good lab. They said, you Americans overblow things.
We do that. On a regular basis.
It's our entertainment.
Got one more. Whose turn is it? I forgot.
I don't know.
All right. Liam writes, hello, TWivers. Have you seen this? He gives a link to a fellow who makes glass sculptures of viruses, Luke Jerem. Yep. Luke Jerem, and absolutely, it's beautiful. Beautiful sculpture on the homepage there. In fact, way back in TWiV 50, Jason Rodriguez, it was his pick of the week.
There's a problem.
What's that?
He should have talked to me before he did the smallpox guy.
It's wrong?
Yeah.
Why don't you email him and tell him?
I hate to, you have to make it all over again.
Yeah, but it's art, he could be wrong.
Okay. I can send him it's art. I can send him the link to vacciniamodel.com. Tell him, melt that sucker down, start again.
Well, I think probably, yeah.
Well, or just tell him, if you're ever planning on doing this again,
check this out.
Yeah, we had this in TWiV 50 a long time ago. I think it's great that he does this and brings more awareness to people.
Oh, they're beautiful.
It's pretty nice. It's really
they're gorgeous.
Absolutely beautiful.
So, doesn't hurt to put them again. All right, let us pick some science objects of the week. Alan, what do you have for us?
Well, my pick is actually something that I know we've picked previously on TWiV, way back on number 23. But I finally got around to reading this book. It's Autism's False Prophets by Paul Offit. And am I the last TWiV person to read this?
Yeah, I read it.
No, I haven't read it.
Oh, okay. Well, at least I'm not the last one. And this is Paul Offit is a scientist and physician who developed the rotavirus vaccine that's now used around the world, and has been very outspoken about the whole anti-vaccination movement, and has received death threats as a result of that. But this book delves into the history of not just Andrew Wakefield, but the other folks who have scammed and exploited the parents of autistic children. And it is, I was familiar with the outlines of most of this stuff, but I wasn't familiar with the details. And it's really eye-popping. I mean, it is amazing what has been foisted on these people mostly in the name of money. And it's a ripping good read. He's a good writer. And I think I read about halfway through the book in one afternoon sitting on the beach in Puerto Rico, and then I finished it on the plane home. I highly recommend reading this book, even if you think you know about this story.
All right.
Yep,
it's my turn.
It's still in the TWiV bookstore, so
Great.
You guys can find it there. It is really good. Yeah, you got to do that, Rich. Then you could pick it again if you want.
Yeah, okay.
Does the TWiV bookstore also give people the Kindle link?
The Kindle is not available for listing in a Kindle bookstore, unfortunately.
That's too bad.
They want to keep all the money for that for themselves, I presume.
Okay.
So anyway, I don't know how many people are buying. I rarely see any activity, but if you just want a place where you can find the link and then go buy it on the Kindle, check out the TWiV bookstore.
Yeah.
It's there. It's always in the show notes. Rich, what do you have?
So I have something that's a bit off the wall here, but it had great relevance to me last week when I was on a little sailing expedition on the Florida coast. This is the National Oceanographic and Atmospheric Administration's National Data Buoy Center. So
Which I already had bookmarked, by the way. This is a great site.
I was looking around for a TWiV pick this morning, and I realized it was sitting right there on my browser. Okay, which was this. This is NOAA's facility for collecting and tracking data on hundreds of buoys, both weather buoys and navigational buoys. Well, no, I guess they're mostly weather buoys worldwide. So there's a world map with all of these buoys on it. You can click on any particular buoy and get data off it at any particular time. So sailing, I use them to understand what's going on in terms of wave heights and wind speeds and water temperature and all the rest of it. But it's just sort of interesting as supplemental data to see what's going on. When you click on one, you actually get a picture of the buoy. It'll give you graphical history and everything else. And so I can spend a lot of time just messing around with this.
This is also extremely useful for scuba divers and fishermen.
Okay.
Because it gives you, a lot of them will give you water temperature at the surface. Some of them will give you water temperature a few feet down, too.
So one of the things that I discovered playing with this morning is that there is a subset of these buoys that comprise the Tropical Atmosphere Ocean Project. And you can see them, in particular, scattered out between the North Pacific Ocean and the South Pacific Ocean in these vertical lines. And this is a system of buoys that are set out there to basically look at changes in water temperature at a variety of depths, and they're used to track El Nino and La
Nina and El Nino.
Yeah. Okay. And so that makes for an interesting site as well. At any rate, there you go. It's fun to play with. A lot of data. Of data.
Cool. There are none by me down at the Jersey Shore. None of those buoys.
Is that right?
Yeah, but there are other kinds. I learned about buoys. I had to take a course to get certified for sailing last summer, and it told us all about different kinds of buoys. But there aren't any of these where we sail down there. They're just local things, but it's pretty cool anyway.
There are there's one off Long Island that would be relevant for you, I think.
If I ever got that far, but
but it's the same. It's all the North Atlantic is pretty much the same from Long Island down through Jersey.
Yeah, right.
So you're in northern New Jersey, right?
No, I am in. I'll tell you where you go on this. So I went onto this map. I clicked USA Northeast.
Okay.
And then you zoom in on New Jersey.
Right.
And then you'll see the town that's most relevant is Seaside Heights, say. It's around there, and there's nothing in that area. But as Alan said, there's one up in Neptune, which isn't too far north. There's a red
I see
one. Neptune is
Township.
close by.
So that's too far north for you?
I wouldn't sail up that far. So you see how there's a barrier beach along the coast?
Yeah, right.
So we sail inside that, but we don't go out in the ocean.
I see. Okay.
We stay in the bay. That's a bay with shelter. That's easy.
What kind of boat do you have?
We bought last year, actually, a a Bahia. It's a laser Bahia. It's a 15-foot kind of
nice
a racing boat. It has a mainsail, a jib, and a spinnaker. Yeah. And this summer we're going to put up the spinnaker.
Excellent.
My two boys took sailing lessons for a couple of years. They're pretty good at it. And my older son wants to go really fast.
I don't blame him.
So he wants to put up the spinnaker. And it's really fun because they work. The younger guy, Devin, is the crew, and he does the jib, and he handles the jib and all that stuff. And Aiden barks out commands, and I sit by and watch, and it's fun.
Awesome.
Yeah. All right, my pick is actually a website by one of our listeners, Jim from Virginia, who is always sending us email.
Yes.
He has started a site called Media Mining.
This is his site.
It's his site. Curated knowledge for innovators is the subheading. And what he does, he loves podcasts. He listens to like 60 a week, he told me. And he just writes summaries of the ones that he really likes, 1-paragraph summaries. So Media Mining 16, March 3rd. And he's got a bunch.
Cool.
There. And now and then he'll have some TWiV and TWIM and TWIP summaries as well. But he actually... After I had picked this, he wrote me an email while I was away, and he said it's too much work. He's got to make them shorter because it's taking time away from listening to podcasts.
There's a lot. He's got a lot of stuff in here.
But it's pretty cool. If you want to go, he's curating it for you, which means he listens to a lot of stuff, and he puts the good stuff that he thinks you'll like. So if you want to find other podcasts, it may be good. So give Jim some traffic.
Great.
Because he's a good fan of us as well. And we have 3 listener picks of the week. Mark writes, "Hi, TWivers. I love your podcast. I am a postdoc in Joe DeRisi's lab at UCSF, and I know that right now I am supposed to be aiming for a faculty job, but my real goal is to discover something cool enough to end up on TWiV."
Those are not mutually exclusive goals.
You don't have to be that cool to get on TWiV.
How about your work? We could just discuss it, Mark, Anyway, the main reason I'm writing is to suggest this pick of the week, a fascinating fresh air interview with Craig Timberg, the author of Tinderbox, A History of the HIV/AIDS Epidemic. So he gives a link to the website and the MP3. Thanks for TWiV. It is really a very good thing.
Great.
Cool.
Henry writes, TWiV team T to the second power. You got it, Rich?
I got it.
All right, T squared. This one is primarily for Rich since he suggested Battlestar Galactica. I lost a week of productivity with that one. Thanks a lot. Rich, people will listen or watch to what you recommend.
No. Well, Battlestar Galactica is, shall I say, a worthwhile waste of time.
This one is, I presume, a television show called Regenesis.
Actually, it's a TV show, but he's got this is online access to it.
Hulu.com, yeah. I had watched the first season while I was in Iraq as a medic. It is pretty good biomedical sci-fi, though the language and content make it for adults over a general audience. I do not have any questions at this time. I have listened consistently ever since I joined a virology biochemistry lab for my PhD work. Thanks again for the great show. All right, one more from Rick, of course, was this Biopunk Do-It-Yourself Scientists, which we read before. So that's our third pick of the week. And that will do it for TWiV 174. And, of course, you can find us on iTunes and at microbeworld.org. You can stream us to your iPhone or Android device with the Microbe World app. Check it out. If you're new to TWiV and you like it, leave a review over at iTunes. It helps us to stay visible, and that way more people discover us and we grow the show. This past week, our last podcast was number 1 for a while in downloads. So cool. Thanks to everyone for doing that to us. And, of course, don't forget your questions and comments should go to TWiV at twiv.tv. Alan Dove can be found on the Twitter at Alan Dove and at alandove.com. Thanks for joining us today, Alan.
Always a pleasure.
And Rich Condit is at the University of Florida, Gainesville, and also vacciniamodel.com. Thank you, Rich.
Quite welcome. Enjoy it every time.
And I'm Vincent Racaniello. You can find me at virology.ws. Speaking of that, I wanted to tell everyone I took a taxi to the airport this morning because it was very early. over in Utrecht. And the cab driver was very talkative. And he wanted to know what I had been doing there and what kind of conference and if I reported my work. And I said, I didn't report my work, but I report on a way to teach the public about viruses and microbes. And he got all excited, and he wanted my website. He said, give me your card with your website on it. I want to check it out. So I gave him virology.ws.
There you go.
And he said, I'll see you online. So who knows? Maybe he'll send us a question someday.
Great.
Got to spread it virally. So I got to make some cards that I can just hand out to people.
Yep.
It'd be cool with a big virus on it, but, Anyway, I wanted to tell you that was pretty cool.
Cool.
You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.
I just got to tell you guys this. I'm going to do about one of the coolest things I've ever done in my life tonight. Me and 3 other guys in a quartet are going to sing the national anthem at the baseball game here.
Oh, awesome.
The college baseball game?
What?
It's a college game, right?
Yeah.